Hello, my name is Dr. David Hartz, and I want to welcome you to these two sessions on the FCA Online CE education. I am really excited about this information, and I believe you will also be as we share with you some science-based information on research that's been around for maybe a century and has been really suppressed because of what it really brings forth concerning what you and I do each and every day, trying to treat people naturally without any drugs and surgery. I believe this information and this science that's coming forth more and more on biophoton physics and quantum physics is going to be the cutting edge medical technology of the future. And I'm excited that we can be one of the leaders in this. And so therefore, over the last several years, I've researched this out in an effort to try to find out exactly how it works. It was evident to me as I used different kinesiological techniques, energetic techniques, that there was something to this. I was getting information that I couldn't get anywhere else from just blood tests alone. And I wanted to know why. I wasn't satisfied with, as many of you guys I'm sure isn't, satisfied with just asking your patients to trust you. So we started researching this, and we found out that there was a tremendous amount of really good information out there. And good, solid research with really fantastic researchers, very well-respected people in the world, which I'm going to share with you, that explains all of this. And so we went through and we've been actually sharing this at the FCA conferences, and we've been asked to put this on the online CE. And so that's what this is all about. And I believe you're going to be really excited as we go through this. Now, what are we going to cover? We're going to cover in these two sessions the challenges of a modern nutrition practice and some solutions. I mean, what are some of the challenges? And there are some challenges to nutrition practice, but I tell you what, it is one of the fulfilling parts of my 40-year career in chiropractic was when I started using functional medicine combined with energetic biophoton testing, with blood work and energetic testing, science-based energetic testing, and then going after stuff I could not touch just with chiropractic alone. I mean, I love chiropractic. It's helped a tremendous amount of people. I treated almost 25,000 people in my practice career, and I helped a lot of people. But when I started getting involved with this, it was so fulfilling. And I have people, even though I'm retired, still knocking on my door and calling me, trying to say, nobody else can help me with these problems. And so I was driven to kind of develop a system. And this particular two sections is, just two little small sections of one module of 7 of almost 24 different sessions that we have on DNS University, Design Nutrition Systems University, that we're going to share here today. And therefore, the types of functional practices and their strengths and weaknesses we want to talking about some proposed neurological models of how kinesiology can work. I don't know if we have this totally down, but I think we got some good proposed neurological models on how this all works, and then taking the mystery out of energetic testing. I don't know about you, but I know that there's something to it. I'm getting great information, but I wanted to know why. And there's science out there and research that really gives us a good information. It just hasn't been shared with the profession very much. And the science of biophoton physics, its history, current research, and how we can use it to get results no one else can. That's the bottom line. It's not just having energetic testing. It's what can we do with it? I mean, if there was ways I could get information totally by blood, totally by functional medicine, at an economical way that people could afford it, I would be happy to do it that way because that's the norm accepted way. But when I found out that this energetic testing was so integral for me getting really good results, then I couldn't just shove it aside. I had to figure out how it worked. So now when we got some idea of how it works, now the key to it is educating our patients. The cool part about this is it puts us in a position, as I'm going to talk about, of being very unique and very sought after because nobody else is going to be doing it. That's what we want. We want to be sought after. So, let's just talk about this a second because, people always talk about everything has to be science-based. Everything, you're not science-based in clinical nutrition or in energetic testing or in chiropractic care for years and years. Well, that's true. And also, we need to widen out our scope of practice in chiropractic. One of the reasons we're fighting for very small, 12% of the population. Okay, it says here 12% say they saw a chiropractor in the last 5 years. 25% say they saw a chiropractor in more than 5 years ago. So we're hitting a very small part of the population because people think that we're just back doctors. I mean, a lot of people think we're glorified massage therapists. And of course, we know we're not. And we know that we have much more education than that. But their perception of that, it needs to be changed. All right. And it's slowly being changed. But there's a whole gigantic group out there of people that are experiencing chronic disease that no one is touching. And I propose that we go after that group. Okay, we say 60% of the males, 25% of the females want to talk to a chiropractor about general health and wellness issues. That should be 100%. Okay, 7 to 11% of female chiropractors are the first healthcare provider they want to talk to about their health. They're not talking to us first. And in my practice, when I developed this system and did it, people were coming to me first. Now, of course, if it was out of my realm or out of my scope, I would send them to the appropriate physician. But because we were having very unique looks at the body that nobody else, they got tremendous amount of confidence in what we were doing, and we become almost a primary care provider. I did not accept that role. I said, please keep your primary care provider, but we kept them very, very bored. The primary care providers saw these people, they were healthy. We were helping them with things that they weren't, and the need for their medications and the medical intervention decreased tremendously. And so 80% are dealing with some kind of chronic disease and want to avoid them. And listen, standardized medicine, first of all, is not addressing the underlying cause of the chronic disease. And secondly, they're certainly not doing anything to prevent it. I mean, they're just waiting for symptoms. Then we give them chemical patches, which, of course, we know has tremendous amounts of side effects. So that's not working. And so also we want to be involved with the motivators of healthcare that prioritize spending. And I mean, we want cash practices. I mean, that's what I want. I'm tired of messing with insurances and having to be controlled by them. I mean, if it's all fine and dandy, if it gets better in the future, but I don't think it's gonna. I mean, I went and practiced from where we just send an insurance form in and they paid it to where you couldn't get anybody to pay it. And I don't think it's going to get any better in the future. They're going to have standardized payment according to what they consider your results. All right. It's, results-based payment. And unfortunately, it's not going to be so much results, how much you help them. It's going to be how many visits you release them after. And so you're going to end up releasing people before they're actually helped. They're not going to be happy with it. Medicare is putting out a whole website now that rates you with stars, and people can go look on there. And unfortunately, the rating is how many visits you give people. And so if you just treat them one time and release them and don't help them, you'll be rated really high. I mean, what good is that for anybody? So we got to get out of this thing. We got to be getting something that I believe is the cutting edge technology that people will pay for out of their pockets. But there got to be something that's so strong that they will pay for it. I mean, and I believe we get there by trying to get to the power of diseases. If we're treating little wry necks and pain in the back and so forth, people can take drugs and cover those over for a while if they really have to. In a crisis of a downward turn of the economic conditions and whatever else, people will start cutting back. And when they do that, if we don't have something that we're treating that they can't, avoid getting the treatment for, or they can't replace somewhere else, then, they're going to use something that's being paid by their insurance. And so the chronic inflammatory-based diseases, we know it's IBS, autoimmune disease, Hashimoto's, GERD, hormonal dysfunctions, all the ones we see here, ADHD, autism, this stuff's going off the chart. Dementia is increasing, psoriasis, eczema. People are breaking out with rashes and stuff all over their body chronically, and it's an upregulation of the immune system, and nobody's knowing how to handle this stuff. Chronic inflammatory bowel disease and allergic rhinitis. All these, there's almost 200 chronic inflammatory diseases. These causes are not being addressed. Okay, they're just covering them over. They're suppressing their immune system with biologic medications that just suppress the immune system. And we're not getting to the cause of the problems. And so also people want to age healthy. If you could just take a population and say, listen, we can look at what's going on, genetic markers as well as indicators that we can find. We can straighten things out in your body, and we can give you a program that will decrease. We can't guarantee we'll make them live to 120, but we can decrease the chances for chronic disease, keep them off some medications. I mean, people come from all over the place for this. I mean, they're willing to pay for it, especially if it's reasonably priced. And so that's what I believe is the key to all of this. And then alternative practitioners are uniquely positioned to do this. as chiropractors and as naturopaths and as acupuncturists, we're uniquely philosophically trained to think of a holistic natural approach. Medicine just isn't. They fight this in their brains. They just want to find a symptom and give a remediation for that symptom. They want a corollary. You got a headache, I got a pill for that. You got inflammation, I got a pill for that. You got, inflammation in your bowel, I got something that just relaxes your bowel. If you got spasm in your bowel, I got antispasmodic. I mean, they're not trying to find causes, okay? And then they have medical standards. I mean, I have so many medical practitioners say, I'd love to be able to do this stuff, but when I do it, I have these medical standards of care where they can take my license from me because, I'm not treating it according to their medical standards. Well, in natural health care, especially in Florida, we got one of the best laws that basically says we can treat people, diseases of the body, with natural means. And so our medical standards of care are exactly what we do. Alternative health care, which is fantastic, So we can do this and really, really help some people. And functional medicine practice, I believe, is this piece of this puzzle in the healthcare that fits perfectly. Okay, I call it functional nutrition. I don't call it functional medicine because I don't think we need to get into that bailiwick because there's too many people now doing functional medicine. They're doing nutraceuticals and they're just treating people symptomatically, and I think that whole terminology is not going to do well. I think we need to brand it as functional nutrition and then make it very successful and very effective. Okay, so let's talk about a little bit of the types of functional nutrition practices. I'm not going to spend a lot of time on this, but I think this is important because it's out there and it's making a difference. All right. So the first one is a practice management model. I call it the high dollar case fee. This is mostly done just with blood and maybe maybe a little bit of symptom survey forms running off of blood only. The pros is you get less volume of patients to see. You don't have to worry about many patients because you don't get that many because your fees are high, but you make a lot of money off each one. And so therefore, you can get good potential income. You don't have to deal with a lot of folks. The con is considerably less conversions. In other words, you could have a workshop where you have 25, 30 people in it. You may get 2 new patients out of it. Some of the other models I'm going to show you, you will get 95 to 98% of the new patients out of it. And so it depends on what you want to do, but you end up with less referrals, you need a higher income base population to handle it because with the functional blood, you're going to have, $3, $4, $5, $6, sometimes $1,000 programs with the blood and some beginning care. And therefore also it drives down your number of new patients because you'll have a reputation of needing that kind of money to see you. So if you have a real high income population, a lot of people there can pay it. That may work for you pretty good. Unfortunately, it doesn't work in where I was practicing because there's just a lot of medium income people. And you need more sales skills because you got to increase the compliance. You got to kind of sell them a little bit more. And I was never really good at that. I didn't like that to myself. But if you're really good at that and that's the kind of practice, that's great. And it's got to be driven by advertising. You got to derive a lot of people to get in front of to get those small percentages of them converting. So you got a lot of high advertising fees to do that. So that's model 1. The model 2 is a lower fee, higher volume model. You see more people, but you end up getting a lot more higher, lower fee, but you still make a decent living. The cons is the challenge of handling more volume patients. You get a lot more people in your office to handle. You got to see more people. I mean, I like that because I like seeing a lot of people, but it's different for everybody. There's a low commitment level with this because they're not shelling that, $5,000, $6,000, $7,000 for a program of care, so they don't have as much invested. But I found that if it's done correctly, you lose next to nobody with this. The pros, you get increased new patients because the financial bar to get in the office is lower. And you can see I'm an advocate for the lower fee, higher volume practice because it works better. It's so much less stressful. And people come from everywhere to get it, and it's a fun practice. Practice stability with increased numbers, if you get a little drop here and there, or also if your economy drops a little bit, then you don't get hurt very much because it still doesn't cost a lot of money to come. And plus you're also helping really stuff nobody else can help, so your practice doesn't get affected. It's more economically stable. So then the clinical practices are this. You recommend nutritional from symptom survey forms, nutritional software, generally nutritional needs. Can also use blood analysis to do this. Then you also, there's another type where you do evaluations containing a more comprehensive entrance form and nutritional software. It's a nutritional-based examination and blood analysis. So you add a little bit more in. You do a comprehensive entrance form, you have maybe a nutritional software, and then you have a nutrition-based examination. So you can kind of, a lot of people do this by slowly putting this into their practice, okay? And this is good. You can help some people with this. The type 3, though, is kind of what we suggest and we utilize, and there's nothing wrong with the other ones either, is we use a comprehensive 26-page history and then a conventional nutritional exam, in-house urine testing, oral testing, ANS evaluation on HRV is helpful, not necessary totally, but it's helpful. Kinesiological biophoton evaluation-based exam, which we're going to be talking about this, and then the functional laboratory analysis. We put that all together with blood, stool, and saliva, and one complements the other, along with the 26 page. It sounds like a lot, but people actually love it. They gripe a little bit, but they're really impressed that you're taking the time to look at it, and you get tremendous amounts of information. So the more comprehensive you are, the better results you're going to be, and the more your patients are going to be very, very concerned or knowledgeable of the fact that you're very concerned about them and are looking at things nobody else is looking at. And then you go get all their blood work they've had before. You take a few minutes and look at all that. Then you can come back with a really comprehensive report of findings that really zeroes in on the causes of their problems. So we have evaluative tools. There's symptom survey forms. There's nutritional-based exams. There's functional blood analysis. There's GSR. Sometimes you can get machines where you can hold different types of grounding and put a small current through you and hit you with frequencies and see how your body reacts on a galvanic skin resistance. Those are all helpful. I don't think they're as good as good energetic testing done the way we do it. I've had all those machines. I've tried them all. I think they're beneficial to a certain extent, but they have some flaws in them. And then a biophoton evaluation where we actually use the emission from the body, measuring it quantitatively. I believe that is, I've had the best results with that compared to anything else I've tried, and I've tried everything there is, I think. So why do we use neurokinesiological and energetic testing? The reason is it's with some of the people wondering how it works and some of the inconsistency that some people have. It's because of specific detection of underlying chronic problems. It gives us information we just can't get any other way. We can't get from the blood. Specific testing of a remediation to identify organ tissue. In other words, we can actually test exactly what works on a specific area of dysfunction. And when you get blood, you'll get, say it's a fungus. Well, you might have 4, 5, 6 different things that you can just throw at people. So what you end up doing is you just try something, throw it against the wall, and then hopefully testing 3 months later with the blood, and then hopefully you hit the right thing. If you don't, the patient's usually gone. They're not going to wait around. I mean, you lose them quickly. With this, you're lasering, as I'm going to show you in a second, and then people get better quickly. Identifying the most appropriate and effective intervention the first time. Identifying allergic reactions. You can get some idea whether or not the body is going to have an adverse reaction to it with a certain percentage of what you give to avoid those. And they get a reaction to a clinical nutrient, chances of them quitting is pretty high if you haven't told them ahead of time. Specific dosing of each individual. There's no way to dose it. You just kind of guess and clinically give it. With this, you can actually tell by measuring the individual body exactly what they need, and keeps the cost of evaluation and treatment down because you're getting a lot of information from the energetic testing. The functional blood is kept with a realistic. I have a really good functional evaluation lab-wise. You can do it for $100 to the patient with a functional lab, report that comes at no charge, and then you can add in, the Cyrex and the Genovia and all the other more complex stuff, and we know how to do all that and we teach it all. But most patients don't need it because between the basic and the energetic testing, we get the information we need, but we have that all, and then we give it when it's appropriate, and the patient doesn't mind paying for it at that point. We don't have to do that on everybody. So why do we do why do some of the most effective functional nutrition practices use kinesiology and bioenergetics? It's results. That's the only reason I use it. I'm not interested in pushing on arms and do kinesiological testing if it doesn't work. And the reason I do it is because I get information I can't get any other way. Okay, that's the bottom line. So the concerns of kinesiological and bioenergetic testing is subjective muscle testing, not reproducible. That's what I was concerned in the beginning. I want something reproducible and I want to be able to be sure that when I give somebody something, they really, really needed it, It seems unexplainable to the patient. Well, we got information now to explain this. We got much more scientific basis behind it. Not wanting to appear mystical. I don't like being strange. I like being mainstream. Everybody wants to be mainstream. Well, the only way I knew to do this, since it seemed to have such good benefit, I couldn't get rid of it, was let's bring it into the mainstream. Let's get some of the scientific basis behind it and make it, instead of being mystical, into the cutting edge. And that's what happens when you teach this to your patients. They go, wow, this guy is just way ahead of the curve. And when they walk out and tell people about it, they have something to tell them. Before I got all this information, people coming from 3 different states to me. There's people coming just trusting me. But my goodness, if I could have had the science behind this and something like books and so forth to give people and explain, videos and stuff to explain how this works. Instead of having somebody going out and referring in maybe 10% of the people they told, they've been 90% of the people. We could take over towns with this stuff, and that's kind of my vision for it. So the dependence of clinical results, only a lack of scientific basis behind those results. And so we need to make the science-based. And the stages of new ideas. This is how it always happens. The first stage is skeptics confidently proclaim that this idea is totally nuts. It violates all science, can't possibly work. Did this with chiropractic care. They're nuts. There's no way they can do any of this stuff, And then the second stage is, well, maybe they concede that the idea has possibilities, but it's not very interesting, and the claims that they say that it works is very weak. Okay. And then the third stage is, well, starts to be a little more mainstream, realizes that not only is the idea important, but the effects are much stronger and pervasive than previously imagined. This could be really, really good. And of course, the last stage is, we achieve this when the same critics who previously disavowed any interest in the idea begin to proclaim that they thought of it first, which is what happens all the time. Happens in medicine all the time, especially with chiropractic care. And, we thought of this first. It was really our idea, and it was, and they just take it over. They come in, try to take it all over. They don't give credit for where it came from or make a place for that person, that place, that profession or idea or the basis of it being something that's very helpful, make a place for it to exist. They just want to take it all over and say it's theirs. And that's what medicine does. And I believe they're going to do this with this technology if we do not come in and claim it and start educating the population on the fact that we're the ones that actually should be the forerunners of this. Okay? Now, let's talk about standards of care just a second, because everybody wants to say evidence-based care. It needs to be evidence-based, evidence-based, evidence-based. Well, let's just talk about this a second, because medicine states always that chiropractic is not evidence-based, and that theirs is, and that, of course, certainly energetic testing and all this is not evidence-based. But the British Medical Journal did a study on a couple family practices, and they watched them over a period of time, and they took every single thing they recommended and went back to the literature to see how much in a medical practice the recommendations are evidence-based. And they found out that it was 18% of all medical recommended care is evidence-based. So this is a profession that tells us that they have all the evidence-based care. Now, does that mean we don't continue to be evidence-based? No. But it also means that, they're calling the kettle black here. They're saying that they are, and they're not themselves. And this is the way this thing works. You have scientific politics. You have, the new theories of research that basically comes out and starts to be researched. And then you have the refuted political science that comes along and says, that's not any good. And we're going to talk about why this happens with this, because there's a big reason why they're fighting this, and has to do with pharmacology. But they come out and start to refute it and say no. So a research project comes out and says this is why you see so many contradicting research, because some people come out and say, omega-3s are great for preventing cardiac arrest. And all of a sudden no one come out and say, no, it has nothing to do with that, and that, it doesn't work. And then you start looking at the people who do the research, and a lot of them are financially backed by the statin manufacturers and things like this. And this happens all the time. I had a friend that I knew of had an NIH grantee for doing research on about 3 or 4 Chinese herbs to suppress cancer. And he got like 65% remission of a specific cancer by these Chinese herbs. He couldn't get it published anywhere. Great research. And when they asked why, he said, we're just not interested. And that's correct. They're not interested in something that they cannot produce a pharmacology from, that they can make a lot of money from, So they're not concerned about natural remedies. It's just not in the big flow of the dollar bill, okay? And so you have to get through the refuted political science. So you have all these different people on medical boards of the supposedly, quote, important journals in America. And if you have to get into those journals to be published and then have it be a couple times and gone through that process before it be accepted as settled science. Well, when you got people on the boards that are controlling everything that comes in, then it's very hard for anything to become settled science besides standard pharmacological types of research. So they did a study, another British medical journal. The members of editing boards of the major journals received up to several hundreds of thousands of dollars in contradictions in conflict with their own interest policies. They have policies that says, I can't do this, and 50% of them receive some form of payment. And so when they go into these boards, they receive payments to control what gets published. And then when they come out of there, many of them will go to work for the pharmaceutical companies for, six-figure salaries that pay them back for their being, cooperative while they were there. And by this, they control everything that comes through. And so you end up having to look throughout different parts of the world into different research, because in America, we're just not going to do it. We're just not going to do it, okay? And so let me just give you some quick examples of scientific politics that occurs and has happened in the past. We got William Harvey, in the 17th century, English physician, who was the first to describe blood circulation to the heart. This guy figured it all out. He said, the heart actually pumps. It goes from there, pumps out throughout the entire body. Well, guess what? There was another guy who said it came from the liver, all right, and that it was the liver that was producing all the blood. And so that guy actually was Galen, and he is a second century philosopher, and everybody seemed to like this guy. And so Harvey's findings was ridiculed, and this guy was ostracized and became a recluse and just absolutely destroyed him. And then it was, 1,500 years later where they discovered, yeah, he was right. Okay. And this happens over and over. Let me show you. In the genetics, Mendel's genetic theories. This is the guy that first came up with recessive and dominant genes that we know today to be the correct. He died in 1884, but it wasn't until several decades later, in the beginning of the 20th century, that his ideas became modern genetics. He was totally correct, but of course, maligned and ridiculed for his, theories that were way ahead of time. Another guy, Ingrid Semmelweis. He was the guy who it was people were doing autopsies and then not washing their hands and going delivering babies, and these ladies were getting postpartum infections and dying. And he came along and said, hey, I think we're transferring something here. And he started taking chlorine water and having people in lime solutions and washing their hands before they did that. And he dropped the mortality rates down to 1 or 2%. And he said, listen, there's something going on here. Well, guess what? He was rejected, and it drove him to alcoholism. The guy was rejected. He was kicked out. He said he was nuts. He said all of his theories were total craziness. And of course, later on, Louis Pasteur came and discovered that he was exactly correct. And of course, how many more people would they have saved lives if they were just listening to this guys earlier? But he ended up in a mental institution where he died forgotten by his peers because he was so rejected for his ideas that were ahead of time. This is William B. Coley. This guy was paved the way for modern immunotherapy. He started injecting and stimulating the immune system to fight cancer cells. And he died in 1936, and his work was totally surpassed by chemotherapy and radiation, because that was what was being developed at the time. And he was one of the first persons. What is the most cutting edge thing we're doing now for cancer? Getting the body to go after the cancer cells by its own immune system. How far along would we have been if they would listen to this guy instead of trying to sell, radiation and chemotherapy and not trying to stimulate the body? Of course, we're still trying to do that even today. And then Francis Pétain-Rousse is another guy who he basically said that viruses played a role in transmission of specific cancers. He wasn't rewarded for that for 50 years. He was ahead. So what I'm trying to say is that there's all kinds of different types of ideas and theories that are not accepted when they first came out. And I believe what we're going to be showing you is this. Now, as we know in functional medicine, we have causes of inflammation and chronic disease, which are chronic infection, toxicity, nutritional deficiency, dysfunctional biome, and food allergies. In our system, this is what we go after. And the problem is, how do we go after? Listen, we have all these different things that can affect, say, a chronic virus. There's a number of different things here that we could give for that. Which one do we give? I mean, that's the problem. Which ones do we give? Here's something for possible parasitic involvement in the body, chronic infections. Which ones do we give? We can choose from it. We just start giving our favorite ones. Maybe we'll hit it, maybe we won't. With interject testing, we don't have to do this. Clinical nutrition intervention for fungus. We have all these different things. Which ones do we give? I mean, these are good things. They work great. If you happen to hit the right thing for the right thing, it works well. Here's toxicity. We got all this stuff we can give, okay? But how do we laser the protocols? How do we get them exactly matched up from individual person to person? The only way I know to do this with this energetic testing. And it does do that, okay? So let's go through and talk a little bit about the neurokinesiological testing, because until we get some machines, which some are being developed even in Russia that I'm looking at, but they're just not to the place where I think, I could recommend them. Also, the kinesiological testing, you like it or not, when patients get it, they feel it. And it's a tremendous feedback system to them. And so let's talk about this just real second. The characteristics of kinesiological testing, it's a neurological challenge in muscle strength. It's actually a muscle lock. And it's pandemic. It has a tendency to affect the entire body. So when we're looking for types of neurological recommendations for how it actually works, we got to think about things that can affect the entire body, all right, which is, a different deal. I mean, I'll show you certain reflex loops we can tell about a certain area of the body, but when it systemically changes the muscle strength of the entire body, it's got to be something else. And so like we see here with this reflex loop, you can have these different anterior horns that are affected, and we all studied this when we were in school, and it can definitely affect the anterior horn, posterior horn of a simple reflex. But we also have some posterior spinal thalamic pathways and these posterior columns, but especially what I want to show you is these down here, because we're going to have to be able to connect the spine reflex to the thalamus and the hypothalamus to be able to get a systemic, parasympathetic, sympathetic, autonomic change. We've got to figure out a way to, how is it affecting the entire body? So we have this reflex loop, and certainly it's going to be affected on the afferents, the efferent neurons that we know come out to the skin and into muscle. And I think there can be certain reflex arcs that affect that. But we got to be able to have something that goes up, because it would only affect just one area of the body if it was just merely this particular level. And certainly when we have somatic referral pathways, we do get a connection between viscera and sympathetic patterns on the surface of the skin. And when we do test over these areas, certainly that can be something that we're stimulating as we're doing this testing. And then there's pathways to the sympathetic neurons. So we have the whole sympathetic chain, the parasympathetic chain, that runs from the lower part of the hypothalamus and the thalamus, and then it exits down, as well as this splenic chain that starts from the gut and goes up, and we of course have the vagus that controls it all. And there is connections from the spinal column to the sympathetic chain that does go all the way up to the hypothalamus. So what I'm proposing here is a model that is actually affecting the hypothalamus and the different parts of the brain that will affect the whole autonomic chain, because what's happening is we're down regulating the whole nervous system when we're getting these changes, because when we get an active reflex, the whole body gets weaker. All right, so there's got to be something going on to do that. And we see these spinal thalamic tracts actually come up through the posterior chains and affects that part of the brain. So what I'm proposing with this connection between the effector organs and the spinal cord itself is a type of chain of a neurological reaction that starts at the sympathetic skin receptors, which we have over our palms of our hands and in different areas of our body, that affects the sympathetic ganglion, that affects the spinal cord, brainstem, limbic system, hypothalamus, back down through the brainstem, spinal cord, through the autonomic ganglion, the sympathetic fibers, to the muscle fibers, the testing muscle. And I believe also energetically this is doing this. I believe the biophoton emission of the body is also reflexing back through the spine, because we can get some changes without even touching the body. And so there's got to be interrelating, which I'm going to explain all this as we go along, with the energetic emissions from the body also. There are some research and muscle testing that's out there. You can look this up on the internet, but just to let you know, there has been some research. Some has been positive, some of it has been negative. I do believe it's because it hasn't been calibrated. There's correct ways to do muscle testing, and if it's done consistently the same way, very consistently, it becomes very, very reproducible. Without that, you can be variability to it. But there are ways that can be tested. In our system, we test it so that it's very, very reproducible. Biophoton and energetic science. Let's start this and tell you a little bit about this, because this is really the most exciting part of what I'm going to share with you. Because there are theories of cell communication that basically are in existence today that do not explain everything that happens in the body. So you have all these cells, and they have to communicate together. And there's about 4 major ways that we have theories that they do that. The autocrine is they communicate with the same cell by just gives these little communication proteins and enzymes that are produced within the cells, are produced within the cell and actually affects the same cell. And therefore communication and information is transferred that way. And then there's gap junctions which occur when you have to have the cells actually connecting together to have that happen. They got to come close proximity to each other. And then there's little tubules and channels that develop between the two cells. And then these communication enzymes and proteins are exchanged and information is exchanged. That's the one model. The second one also is the paracrine signaling, where you have local activity between two cells that come close together with paracrine. So they actually, they transfer these protein molecules can actually interrelate with two cells, but they have to be very close together. And they have to, by random, those proteins have to hit the other cell to become, effective and to transfer information. And then the last one is endocrine, where the chemicals are just excreted out into the bloodstream, and those hormones end up hitting receptors in different target organs and areas of the body. But that's very slow. I mean, all this is very slow. I mean, and plus also the probability of this occurring is slow, and it doesn't account for all the reactions which occur in the body, because there's over 100,000 reactions that occur per second in a cell in the body. 100,000 reactions and actions that occur in a cell per second. And there's no way those particular cell communication theories will encompass, even though they're true, they can't account for everything that happens. What's happening is happening at the speed of light, and none of that is fast enough to account for it. And so for every one protein molecule, there are 100,000 water molecules. So if a protein molecule is released, there's 100,000 water molecules around that could get in the way of it reacting with the receptor that it needs to happen. So, I mean, it just can't happen that way. And so also the chance of interaction at the speeds needed to produce is a serious question. And it's not just misfunctioning chemistry. So this is the bottom line. Medicine wants the body to be one big, gigantic bunch of chemical reactions. That's what they want. They want to believe that we're just, the whole body is just a bunch of chemistry. And when you get sick, it's just a chemical reaction that's gone awry. And the reason is because we deal with replacing and giving you chemistry. And we have a chemical for you that can affect your chemical dysfunction and replace it. As long as it's all chemistry, we have all the answer. If it's all of a sudden energetic, all of a sudden has something to do with other types of communication, we don't have anything for that. And that's, I believe, the source of a lot of the resistance from the very beginning on this. It's outside what they know and they can't control it and they don't know anything about that and they just don't want it to exist. And so, but guess what? There's something else controlling all this. Even Albert Leininger, this is a Nobel Prize winning biologist, basically, in his textbook, which is still the kind of D2 textbook of all people who study this stuff, basically, when it comes down to biology, makes this statement. He says, the body purposely directs chemical reactions by means of electromagnetic vibrations or biophotons. It's one little statement in his textbook, and then he goes off on all the other chemical reactions that can occur. But at least he's honest enough to say that, because there's no way in the world that at 37 degrees centigrade temperature, all that stuff can happen just by the known reactions chemically. It has to have an energetic electromagnetic vibration of something that's orchestrating all this. And so we know now that this is the case, all right? So the human body is a bunch of human light systems. It's a big light system, okay? And it works by light information at sublight frequencies, giving information back and forth. So it's not mystical. It's just early in its discovery and its research. And it's just unknown. It's not settled science in West, but it's researched all over the world, as we're going to see, okay? And so the hormonal energetic mediated is kind of this. You know, you get release of certain things and it just kind of bounces around out here and finally hits a receptor that fits right into this little key, and you get good results, all right? The signaling with energetic believes that there's a signal in energetic frequencies that signals which hormone receptor goes in here, and therefore it's orchestrated by an electrical, but actually in a light frequency, a subtle light frequency, a biophoton frequency that controls literally what happens to these reactions, all right? Now, just real quick on some mitogenic radiation research. Back in 1923, we had this Alex Gershwitz who basically came up with research showing that different roots of different potato plants would actually, if you put them very close together in a glass, that the one little root would stimulate the other root to actually develop into bud, and it was separated by glass, so that was no chemical interaction could stimulate the budding. It had to be electromagnetic. And he proved that there was something being emitted in a frequency from the one bud stimulating the other bud to actually germinate. And actually when the research was done, the percentages were, very, very obvious. And there's some of the percentages. And he put this out. He was, of course, maligned for it. He called it mitogenic radiation. And this has gone down through the ages, showed up in research ongoing, and people followed him and was able to reproduce it. Some people didn't, but they didn't do it exactly the same way. And other people have totally reproduced this through the years. But this is years and years and years ago, back in in the 1920s and 30s, that he first discovered this. And then this guy, the Jane Benavest, MD, This guy was considered for a Nobel Prize. He was the French National Institutes of Health medical director. So the NIH in America, he was the equivalent of the director of the NIH in America, okay? Very well known, very well respected, all right? He actually developed and studied basophil degranulation. If you ever know the ELISA test, which is for food allergies, this guy's one of the first persons to ever develop that. And he was doing research trying to figure out putting small amounts of reactants with antigens and trying to get reactions in white blood cells. And he had one of his people that was working for him come to him. He said, I think I messed up your whole research. And he said, I did the wrong dilutions. I put this very small dilution, extremely small, much smaller than you wanted me to do. But the strange thing, he says, I got a reaction. So he went back and he said, what? And he goes, yeah. He said, I put this very small dilution and we got a reaction. I should have done that, but we did it. He said, well, go back and do it again. He did it again. He got the same results. And he started thinking, wait a minute. So he kept diluting and diluting, and he kept getting more and more reaction. He had nothing to do with homeopathic. He knew nothing about homeopathy, but he ran into this, and he was honest enough to search it out. And so he started doing research. Well, when he started going ahead and trying to publish this stuff, they got so mad at him, said he was nuts. actually kicked him out of the National Institutes of Health, threw him into a little teeny tiny lab, just degraded the guy, and he ended up being an alcoholic, and he ended up being a recluse, and it was just terrible what he did to this guy. But he was really an honest researcher who tried to figure things out. Yes, later on, he came back and was hired by, I think, some people, even in homeopathy, to do research. And he did a study which was unbelievable. If you remember back in lab and when you took, your original studies, acetylcholine and histamine would affect the rate of heart when you took a pig heart, or you took a toad, or something like that, and you actually took the heart out and you mounted it. You could actually put different atropine and so forth, acetylcholine, and you could change the rate of the heart. Well, they did this, but what they did is they diluted histamine and atropine down to the place where there was actually no molecule left of that particular reagent in it, just the energetic signature. And then it interrelated it with the heart, and guess what? The heart slowed down and went and sped up. It was not the atropine, it was the energetic signature that was being imparted into the water from, which freaked everybody out, okay? So they started realizing that there was something in the molecular structure that was an energetic frequency that when we diluted this down to where mathematically there couldn't be any molecule left of that particular, it was still leaving an energetic signature in the water that had an effect, okay? And this guy comes along, Louis-Antoine Monthier, who is the Nobel Prize winner in 2008. He discovered the HIV virus. Very honest guy. He knew Benevest. He knew some of his research. He started doing some of it on the side, and he realized he was right. And he got kind of ticked because he said, you destroyed this guy, and I'm going to try to continue on with his research. And he did. And he did this. I'm going to show you this. This is just unbelievable. He took, and he made this particular little apparatus here where he took an HIV virus vial of actually an HIV virus and sealed it in a vial. And he took another vial and he had just energetically neutral water, and he sealed that vial and he put it in here in this coil. Then he put a small 7 hertz, a little electrical current around the coil. And then he shot a little laser through it too. And he left it in there for a certain number of hours, 18 hours. And then what he did is he took out the vial, okay, that did not have anything in it, okay? And there's something called polymerase, which is used in high definition PCR, which is used in forensics. So when they use it to find people that, do crimes and stuff, like they have a piece of the DNA, they can put this polymerase into with nanoparticles, with just pieces of the original DNA, and it will actually reconstruct it, which we'll see in a second. So they use this, okay? So what happens is this. If you take and you have just the nanoparticles, the basic building blocks of a DNA RNA strand, you put that into the test tube, and then you put polymerase in, which can actually reconstitute it, and then you put just a piece of the RNA and DNA in. That polymerase will take and reconstruct that whole spiral helix from the nanoparticles, the basic building blocks. And that's what got the Nobel Prize for polymerase, because they use this in forensics. If they don't have the whole, just have a part of it, they can actually reconstruct it from that polymerase. Well, what Monthier did is he took that principle and he kind of took it to the next level, okay? And so he took the energetic frequency and he put just the frequency, no molecules at all, into that test tube. Then he put the nanoparticles and the polymerase in there, okay? So now all you have is an energetic signature, polymerase, and the nanoparticles. And then something really unbelievable happened. The polymerase all of a sudden started reconstructing the whole spiral helix of the HIV virus. Nothing in it to reconstruct it except the energetic signatures that were transferred in from that RNA and DNA. So basically what it said is that the DNA was reconstructed 98% totally correct without any of the DNA strand being there, only the energetic signature. So what orchestrates the DNA to be constructed? It's not the particles, it's the energetic input signals that are unique to that DNA. I think that was unbelievable. And so this is extremely important for us to know. He actually took those same energetic signatures, he coded them and put them on a WAV file. He actually put them into a computer and he sent them by email to a French laboratory. They took those signals out. They energetically exposed a test tube of non-energetic water, and it reinstituted that same DNA from those nanoparticles at a 98% correct amount. And so this is something that has been proven to be in existence. Why we don't know more about it is a good question, but he's done great, and he's still doing this. Then Fritz Albert Popp came along actually a little before him, and he was at University Marburg in Germany, and he was researching in a radiological lab trying to illuminate different substances with infrared light. And he was trying to see what happens to these different things when they actually got hit by the light. And he used ultraviolet light, actually what he was using. And he found that he took two different compounds. One was a compound that was extremely carcinogenic, and another compound that was only different by one chemical bond that was not carcinogenic at all. And he couldn't explain, why one was so carcinogenic and only one bond different, and it wasn't carcinogenic at all. And so he hit it with infrared light, and what happened, he found that the carcinogenic molecule took the frequency of light, scrambled the light, and re-emitted it at a different frequency. And the one that was not carcinogenic just passed the light right through and didn't do anything to it. And he got extremely, extremely excited about this. And he did 37 other chemicals that were carcinogenic, some not. And every single carcinogenic one absorbed the light and re-emitted it at a different frequency. So he started realizing that if these cells and their multiplication is being controlled by emission of information by subtle light frequencies, and that carcinogenic molecules are taking these and reformulating that information and then re-emitting it, which could produce an abnormal cell to be produced, maybe that is the key to what's causing cancer. He got very excited, and everybody just basically sent him out to pasture too, told him he was nuts, that can't possibly be so, and told him to go back and show me a biophoton. They said, you're saying all these biophotons are doing this, make something that will show it. Well, he went and did, and he got a research guy that was trying to get a PhD in his department, graduate student, said, make me a photon multiplier that can see all this. And so this really brilliant lab research assistant got his PhD, and he developed a photon multiplier that could see the biophoton emission coming off. You can see it coming off that leaf right there. And actually, they started doing research on how much emission of these biophotons were being emitted. And he did germ capacity evaluation, where they actually could tell how healthy a germination of a seed would be by the emission that was being emitted from it, which is what happens with food. The more healthy the food is, the more energetic. In fact, this is really important because the food that we eat, it's not just coenzymes and vitamins and, different types of sources of nutrients for us. It also has information for how that to be used in our body. So that's why it's so important to have a very biophoton-filled, natural, organically grown nutrient. That's why it works better. It's not just coenzymes and enzymes and vitamins and minerals. It's how it utilizes information, how to be utilized in the body. So biophotons are these little light frequencies that radiate from every cell and everything. And it's a single particle of light. And they actually found that their life cycle is a little bit like a star. It starts out bright, and then it goes through all the way to a kind of like a dark hole. But I don't know what's important about that, but it's an interesting fact. And then I did some research to find out that this biophoton emission is coming out of the spiral helix of the RNA and DNA. This was actually really interesting research that was done by Popp. He actually did ethylene bromide, and he found out that it would unwind the DNA helix when he hit them with this chemical. And when they did that, it caused an increase in biophoton emission. So they know that the spiral helix is actually most likely emitting the biophoton information, and then it's coming out of the body, out of organs and so forth, and we're going to see what it's producing in just a second. It actually it's got a specific energetic frequency that's in about 38 nanometers. It's, of course, above the color range, and it's kind of in an ultraviolet spectrum range. Well, that's actually the end of this first section, and I've got some really interesting things to show you in the next section, and how you can use this to get better results in your practice and to get results no one else can. And so we look forward to seeing you in section 2.